today we're going to look at the extraction of DNA from plant cells using onions. So Carolina, if you could mix the 3 grams of salt into the washing up liquid and add the onion to it, please. Very good. So Jamie, why are we chopping the onion?
It breaks down the cell wall and releases cytoplasm.
Very good. Excellent. So it breaks the cell walls and allows the cytoplasm to be released. Perfect. And Darren, why are we adding the washing up liquid?
To break down the cell membrane.
Perfect. It breaks down the cell membranes. Excellent. Good man. And Ryan, why are we adding the salt?
Minimise the attractive forces between the DNA and the protein, cause it to come together.
Well done. That's perfect. Excellent. So now, Carolina, if you could put that mixture, the salty water and the DNA or the onion into the water bath at 60 degrees Celsius, please. Perfect. That's excellent. Patrick, why are we heating it to 60 degrees?
To break down the enzymes.
Yes, excellent. We're going to break down any enzymes that could damage the DNA, and we're going to leave it in there for 15 minutes. And if we left it in for any longer, it would break down the DNA itself, so we won't leave it any longer than that. Now, we have some that we prepared earlier, so we had heated them for 15 minutes to break down the enzymes. We then cooled it down for 5 minutes. So, Aaron, why did we cool it down for 5 minutes?
It slows the action of any remaining enzymes before they destroy the DNA.
Excellent, good man, yeah. So to slow down any chemical reactions. So Carolina, if you could take out one of those beakers, please, that we've prepared already. Wonderful. And now what we're going to do is we are going to blend it for no more than 3 seconds. So the blender is plugged in beside you, Carolina, and you just want one press. That's perfect. Maybe just one, just one more, just for 3 seconds. That's perfect. Excellent. Now, so Chris, why are we blending?
To destroy cell walls and membranes and to release DNA.
Excellent. And if we don't blend for any more than 3 seconds, because it would break down the DNA itself. That's wonderful. So Carolina, if you could just pour that mixture now through the coffee filter paper, please. Okay. Now, Brona, why are we filtering it?
To separate the DNA from the cellular debris.
Excellent. Okay. Now, so hopefully it will pass through fairly quickly, and what we want to get is about 10 mils of the solution. Now, the solution that passes into the conical flask contains the DNA. The rest of the cell is left behind in the filter paper, so that's wonderful. So what we're going to do now is, we should have 10 mils there, Carolina. If you could just lift the funnel off and just sit it into the other beaker, please. And then you can pour the filtrate into your boiling tube, please. Good girl, that's perfect. Excellent. That's wonderful, that's a lovely amount there. Now, we're now going to add the protease. So you have in the small little beaker beside you there the protease. And we're going to add 2 to 3 drops of the protease. Helen, why are we adding the enzyme protease?
Breaks down the proteins associated with the DNA.
Good girl, excellent. That's wonderful, very, very good. And now the very last step is we have taken some ethanol from the freezer here, and I'm just going to pass it in to Carolina. And you can take 1 bottle of this, okay, Carolina? And what you're going to do is just very slowly down the side of the boiling tube, you're going to add in the ethanol straight from the freezer, please. Girl. So we add 10 centimetres cubed of the ethanol straight from the freezer. Now, Katie, why do we have to add the ethanol directly from the freezer?
DNA is insoluble in freezing cold, ethanol is soluble in alcohol at room temperature.
Good girl. So basically the DNA will not dissolve in the cold alcohol, so it separates it out of solution. And what we're hoping to see, and we can just leave it sitting in the stand there, that's wonderful, at the gap between the 2 layers, so if I just hold it up there for a moment, at the gap between the 2 layers where it looks cloudy, that's our DNA. Okay, so that's wonderful. Excellent. So that completes our experiment for today.